Hi, everyone. Welcome to another episode of the Student Spotlight series, where we showcase some of the work our past students have done. Today, it is our pleasure to have with us Twisha Sharma. So, Twisha, can you tell us a little bit about yourself, where you're from, what year of school you're in, and what course you took with CCIR?
Um, I am like a student from I think a Valley Christian High, and I really like biology. So I joined the program in order to learn more about it and like do research on the subject. So I'm in, I think I was in grade 9 when I joined, but I'm in 10 now. And I was, the research that I did was about like a certain protein called PAX8. And this protein basically influenced like a disease called Hashimoto's thyroiditis. And it was how the variants in the protein influence the disease. So I like try to document a bunch of the variants and like list which ones are pathogenic, which ones are not, and figure out which ones actually affected the disease and which ones did not.
So, just to clarify, was this part of a Future Scholars program or a one-on-one mentorship?
This was one-on-one mentorship.
Okay. So, can you say a little bit about how the program was structured? I guess how did you decide? How did you connect with your mentor, and how did you decide on a research topic, and what kind of process did you go through in writing your paper?
Yeah, so I apologize. I'm a little bit sick right now, but
Oh, no problem, no problem at all.
mhm. So I started off by when I first got accepted into the program, they asked me like during the interview which subject I would prefer to do my work in, and I mentioned immunology because that was one thing I really liked. And then they paired me with um, Professor Dr. Vera, and since then, when I met her, she and I were first just talking about like subjects in like the stuff that we really adored about this subject and what we thought we might be interested in. I like mentioned that I really liked like T cells and stuff like that, although we ended up choosing something different entirely because she ended up going through like possible like immune diseases and like all the stuff about the immune system. And we ended up narrowing down on our own right by saying like, oh, like this interested me at the end of class and like this did not. So we can discuss this more and like this less. So then we and for that process we ended up settling on the disease Hashimoto's thyroiditis. And from there I ended up going through research and during during my research I looked through the ClinVar database. And in that database, I looked for variants that were affected by Hashimoto's thyroiditis. And I just listed like every single protein I could find that had a variant that affected this disease. Then I ended up picking the one protein with the most variants that I could find, which ended up being PAX8. And so all of these, I went through all the variants. I documented them in a PyMol like software. And then I looked at basically like where all the pathogenic variants are. And from that, I like matched the fact that these areas were like considered like N termini and like C termini of the protein. And that's where I ended up doing my research.
So, just as an aside, I do want to put it out there. If you're feeling sick, we can reschedule this. I don't want to force you to talk for, 20, 30 minutes if it seems like you're sort of having some, difficulties.
It's all right.
Okay, cool. Well, we'll keep going. They can edit that out, so it's fine.
Mm.
You mentioned briefly that you talked with your mentor about why you loved the subject as a whole, and I definitely want to talk more about your research, but I wondered if you could say a little bit more about what interests you about biology in general.
So when I first joined high school, I didn't know what I wanted to be when I like grew up at all. And now I know it's pretty obvious to me, but I started off not knowing and I didn't know that biology would be the subject that interests me. Like basically, I thought it was just the mitochondria was the powerhouse of the cell again. And then I ended up um learning more about it started off with cellular respiration and then the cell membrane. And then I realized that biology was so much more than just that. And I was really fascinated about the subject. And so I thought like I really wanted to learn more about it. So over the summer, I read my AP Bio textbook, and that's when I got into CCIR and stuff like that. And because while I was reading my AP Bio textbook, I was fascinated by immunology because I thought it was just cool at first. And then I saw another book on myself that was aside from my AP Bio textbook, and it was called Immune by Philip Detmer. And so I picked up that book because it looked interesting and I didn't know I owned it. And so I started flipping through it. And it was like some of the most interesting thing I'd ever read. And that's what set me on immunology. So that's why I picked that subject for CCIR.
Okay, so back to your research topic. What ended up being the key insights or takeaways once you finished your paper?
Um, yeah. So one of my key insights or takeaways was probably just like in order to do high level research, you don't need to be you don't need to have like four PhDs. You can, if you like know a lot about like the subject that you're thinking about, then you can still do a lot of research on it and still be helpful. You can't just you don't need to like give up and say, oh, like I won't be able to help with this because I'm too young.
I think that's definitely true, but I also meant like specifically about Hashimoto's and the protein that you were
Oh,
researching.
specifically, but we learned about the fact that we found a certain sequence like of proteins that we like analyzed. So basically, there was like some research suggesting that like the pathogenic areas, like the C proteins and stuff, were like the C terminus and the N terminus of the protein were kind of in this one specific area. But like the research, like with the variants, kind of like really helped show that like this is like the one part of the protein that like actually connects to the DNA. Um, that actually connects to the DNA, and like there's two certain parts of it that connect to the DNA, and we helped prove that like these were the two parts because we collected a bunch of data from everybody else and put it in one spot. And with the diagrams, we can look at like what parts are actually affecting transcription and what are not. So,
So, can you say a little bit more about how your work fits into the existing literature? So, you mentioned that you were sort of collecting a lot of data from prior research. I guess what about your approach allowed you to see something that the I guess previous publications on the subject did not see?
um, so a lot of the previous publications focused on one variant, and my research connected a lot of variants. So, I I wrote like 33 variants in particular that I found in the exons of PAX8, and I ended up drawing them from ClinVar and then seeing them again in AlphaMissense. There's two different databases that I picked them from, and then through like an editing software, I like put them into the actual like protein model so that I could see like which areas were more pathogenic and which were not. And while people had like somewhat done like that, it was not with like all the like it was not with a ton of variants. It was with like a very small amount of them. So I could see like more patterns than other people had done so far.
Cool. Yeah, I think that's pretty exciting. I definitely think that it's really important to take a step back and sort of collect existing work and see how, I guess what things might say in combination, because sometimes maybe people don't talk to each other enough. Um, so have you finished your paper?
Yes, it's I'm putting it through publication now. So the final draft is done, but I'm sending it through right now.
Okay. Can you say a little bit more about the publication process? Like where are you looking to have it published?
Um, I was thinking of like the Princeton Journal of Interdisciplinary Research, and it was one that CCIR suggested to me. So I don't really know a lot about journals, so I just trusted everything that they told me and went with that one. And then I reformatted my paper, which took quite a long time. And then I sent it to CCIR again. They sent me a couple of edits I could make, and I'm about to make those edits in today and then send it into the actual journal. And then I'm pretty sure they have to review it, send me a bunch of edits, and then if I end up getting accepted, of course. So
Yeah, it can be a long process. That actually reminded me of a question I should have asked earlier, but could you say a little bit about the support your mentor provided in the sort of writing process for your paper?
Oh, she provided a lot of support. She like taught me how to write the abstract, the introduction, the methods, everything else. Like I didn't know how to write a research paper at all until I met her. And then of course, like the writing style I had to learn, but like everything else, like she taught me with like structure and everything. I definitely could not have done it without her guidance.
Do you feel like the program has prepared you to do research in the future, or do you feel more confident about sort of taking on a similar project again?
Yeah, definitely. Definitely. Now that I've written one research paper, like I definitely kind of know like the structure of them and how the process sort of goes. And so I'm definitely way more prepared to write another one if I had to.
Do you plan to?
I hope to. It was really fun.
Okay, I just it's funny you say if I have to, you know.
Oh, I didn't mean it like that.
No, I know,
I
I know,
just
I know, I know you probably didn't, but no, that's good. So I imagine that you mentioned earlier that in the past you weren't really sure what you wanted to be when you grow up, but now you feel like it's pretty obvious. But you didn't specify. What is it that you want to be?
I was thinking of being like a surgeon. Because I feel like it really interests me, especially dissections in biology in my AB bio class and stuff. They like really interest me and I really enjoy doing them.
Yeah, I mean, I think that if you're someone that likes to do research, being a doctor can give you a lot of flexibility where you can practice and then also contribute to research. So I think that, for a certain kind of person, that can be a really exciting career. Um, so is there anything that you wish you had known earlier or any advice that you would give someone who was going in to do a similar program to the one that you did?
Hmm. I would say if you need to sort of pick your topic kind of quickly, because I especially like managing the work in terms of school as well. Like if you're gonna do it, it would be better to do it in freshman or sophomore year because during junior year you have a lot of work and it's really hard to manage with doing programs like CCIR and writing papers. It's a lot of dedication and you can't just like underestimate how much work it takes to write a single paper. Like that and like of course there's like the time management for sure and then especially like picking your topic quickly because you don't have like a lot of lessons to work on your paper if you spend a lot of time picking your topic.
Yeah, I think that's really good advice. I definitely think it's possible to get too fixated on like finding the perfect topic, and then, by the time you found it, you don't really have enough time to work. So, yeah, I
Mm-hmm.
think that's good advice. Do you have any final thoughts that you'd like to share about your experience?
Um, no, I said a lot. I think I said everything that I did basically.
Okay, well, great. I mean, I'm glad that it was a positive one, and I really enjoyed hearing about your research. And I appreciate you sharing your time with us.
Yeah, thank you so much for interviewing me.